Hello, people. What do we got? We got 22 people out of 35. I guess that's the going average here. Okay, so let's just go ahead and do our quiz today. Hopefully you've found it already. Probably some of you have. So go to exam 3 right here, daily quiz 3.3-29. That's today. Go ahead and do that. You got yourself until quarter after. And I'm out. So if you came in late, we're doing the quiz now. Go to Moodle and open up that quiz, 329. And he's back. Everybody good? I don't know if this thing shuts off at 1:15 or 1:16. It's supposed to be until 1:15. Does that mean it goes until the end of 1:15 or 1:16 starts? Or does this stop at the beginning of 1:15? I don't know. Give it a couple more minutes. Make sure everyone's submitted. Let's see if I do this. What does it say? Quiz is closed. Oh, I'm sorry. The quiz is closed. All right. Let's look at the answers here. Hopefully this was easy. If it wasn't, you need to study more because try to make it easy. Okay, primitive streak formation in the mouse involves multiple signaling molecules. First, the blank is induced in the visceral area, visceral endoderm, that would be G.
We don't see what you're reading.
I always do that. Thanks for telling me. I always do that. Okay. First, the DVE is induced in the visceral endoderm by nodals released by the epiblast. This causes the production and release of lefty as it becomes the AVE. This continues to release blockers, making this the anterior end. The blank then releases nodals onto the proximal epiblast, and that leaves only brachyury. Okay, hopefully that wasn't so hard. Next question. Which transcription factor is associated with the mesodermal fate? That should be easy. We've been talking about that since Xenopus. What hormone is associated with maintaining pluripotency? FGF. What is the change from mobile to presomatic cells called? MET, mesenchymal epidermal transition. What hormone is associated with differentiation? Ooh, that would be RA, retinoic acid. Which transcriptor is associated with neural fate? SOX. Which signaling pathway is associated with somite formation? That'd be Mr. Notch. Okay. Okay. I always make that joke. Okay. It's the oh God, what is it? Kyle and Cartman and that cartoon. I forget. Anyway, neuromesendermal is here. Somite. Oops. Somite. Presemitic is all of this. Neuroectodermal would be the blue ones, and mesodermal would be the red ones. Okay, we've seen this one quite a few times. Hopefully that worked for you. Okay, hopefully we're all keeping up with that. Today, I would like to take the next step. So we've been watching this node regress and cells coming in through. So the next two things to look at is how many somites do we get? You know, what determines how many somites we get? I think I told you on Friday that you know, if this process goes faster, you end up with a lot of somites, and then a lot of body segments. Go slower, you get fewer. So a snake has got a lot versus a chick, which has got fewer, which is us, who's got even fewer. So we'll look at how many somites get formed. And then how are these somites patterned as they go through, as they get formed? They're already patterned when they are in that presemitic section there. And that has to do with the Hox genes. So we'll look at the clock mechanism and the Hox genes today. Yay. I'm excited about it too, guys. Boom. Somatogenesis. Somatogenesis. Sui generis. I've been chatted. Oh, when are we having the exam? Did you decide when the exam will be? I didn't decide. No, you guys did. You made the vote. It's going to be next Monday. Otherwise, we'd just be reviewing today and my life would be easy, but I had to work today because of you. Just kidding, not really. Yeah, but the exam will be next Monday, and we'll cover everything that we get through until Friday. We'll see how far we get in this chapter. I may finish it by Friday. We'll see. But the exam will be Monday. Monday. So no review. Depends on how fast I get through all this stuff this week. Okay. So rearranging my real estate here. Okay, so what determines the number of somites is this clock that runs in this presemitic mesoderm. And this clock involves a bunch of genes turning on and turning off, turning on and turning off. And when that going off cycle goes through this spot, it makes a somite. So I'll make more sense out of that as I keep yakking away here. So here is our presemitic mesoderm. Here's our stem zone. Cells get made here, and here are our gradients. So right here is called the determination wavefront, because as this node moves forward, the gradient of FGF and retinoic acid moves forward as well. And you get to this one point where FGF drops off, so that's below this threshold point that you get differentiation. So that's your determination wavefront. When you get to the point that FGF is so low that it's no longer having an operation, then the somite starts to form right there. So that's why we call this the determination wavefront. At that point, it's determined. Now, what's going to determine how many of these somites you end up with? So if we assume that this node is moving forward at a constant rate, what determines when cells will go through that determination wavefront, or when the somite will form, is how fast do the cells cycle through this gene activation cycle. So let me show you that. Okay, so here's the gene activation cycle that happens. So FGF, those neuromesendermal cells and all the cells moving through that presemitic mesoderm are releasing FGF. So you've got FGF there. FGF causes production of Wnt. Wnt then upregulates Notch. Remember, I said Notch is important for somite generation. The reason why it's important for somite generation is because it produces the transcription factor HARI, and HARI then turns on the transcription factor ephrin A4. This is the key. When ephrin A4 comes up, you get differentiation, okay? So the cells become somites when that gets turned on. So how does this happen though? Why do they cycle? So the next thing is, why are these things cycling? Well, FGF causes production of Wnt, which makes Notch, but Wnt also makes axin, and axin comes back and blocks Wnt production. So now FGF has no effect on these cells anymore until you get rid of axin, and then they'll be competent to express Wnt again. So, and also Notch, when it makes HARI, also makes MSP, which makes this guy called lunatic fringe. Now, these are all transcription factors here. MSP and lunatic fringe are transcription factors. Lunatic fringe comes back and blocks Notch. When it does that, it can no longer do this. Okay, HARI, ephrin, all that kind of stuff. Okay, so when the cells are in this presemitic mesoderm area, they are mostly just cycling between Wnt, Notch, and these things. They're cycling. And it's like FGF brings up Wnt, Wnt brings up Notch, and then these bring down Wnt and bring down Notch. And then once axin gets destroyed in the cell, it only lasts about 30 minutes. When it gets destroyed, then these cells are competent to express FGF. To express Wnt, so FGF causes Wnt, which causes Notch, which then inhibits itself, so it comes down again. So you've got this cycling of going up and going down because it stimulates it stimulates the production of its own inhibitor. And that whole cycle of inhibition takes time, right? This is all the timing has to do with turning on a single transduction pathway, turning on transcription, turning on translation of those new genes. Now those new genes turn off transcription of different genes, and then it all comes down. And then axon goes away, and it can go, and then the whole thing can start over again. So we've got two different negative feedback cycles here that are causing things to go up and down. Now, when you get a somite is when you're at the low level of, or you're at the point of the cycle that ephrin is being expressed as you cross that determination wave point. So ephrin A, what is it? Ephrin something A. Ephrin A4 is high when the cell crosses that determination wavefront. At that point, the cell is no longer competent to, or there's no FGF anymore, so it no longer responds to FGF, and boom, it differentiates. Let's see some animations about that. This is a really complicated thing, so I've got a couple animations to watch here that hopefully are going to help with that. Where are we? Okay, so here's your, let's make it big. Here's your FGF wave. Now this is moving in this direction, right? Here is your node, it's moving down here. As it's moving down, this determination front is moving with it at the same speed. Here's that cycle of gene activation. Now, these cells are not moving with this cycle of gene activation. This is a wave of gene activation that goes up through the presemitic mesoderm until it hits this, and then it stops, and then it starts over again. So let's watch this. Watch, every time you hit the end of or every time the beginning of a blue cycle hits that determination wavefront, boom, you get a new segment. New wave, new segment. New wave, new segment. Okay, let's just watch that again. That's pretty fast. New wave, new segment. That determination wavefront, now. New segment. Ooh, cancel. We'll come to that in a second. Okay, so what then determines how many somites you get is how fast the genes cycle through like that. Okay, in the chick it takes 90 minutes, and that node's moving at a certain rate, so you get like 46 vertebrae or something. But like I said, in the snake, it's going to cycle a lot faster, and you get more somites. Watch this. So this is like a slower, and this is a faster. Notice that the node is moving at the same speed, no matter which side we're looking at. The only difference is how quickly that wave is going. Every time the front blue wave hits the determination wavefront, you get another somite. But there are more waves on the right. Oh, stop already. Keeps wanting me to do that. There are more waves on the right, so you get more somites on the right. And again, one more thing just to remind you, these aren't cells moving backwards. Each cell here is going up and down and up and down and up and down until that cell itself crosses the determination wavefront. Then it becomes part of a somite. Okay. One more video I want you to watch. I don't think I'm going to show it, but I really want you to watch it. Let me see if I can find it real quick. Oops, go away. One. All right, so this one, this is like, it's in your notes. Let me see. Let's get a new share. Come on, baby. Okay, so this is today's notes. This very first animation is this. And this is a woman talking about exactly what I just talked about, probably doing a better job than me. So I would seriously suggest you watch this. And then, and then at the end of this video, she starts talking about combinatorial gene effects. And that's our next thing, because we're going to talk about Hox now. So how do you get, the difference between the cervical versus lower cervical versus thoracic versus lower thoracic? How do you get a difference as you go down the body plan from top to bottom? It has to do with what genes are being expressed in what combinations. And we're talking about Hox genes here. So here, this is like a simple example. It has to do with plants. If you look at the flower of a plant, you've got sepal, petal, stamen, and carpel. Now, I don't even know what those things are, frankly. Stamen I know is the male part. Petal is outside. Sepal must be outside of that. Maybe carpel's on the inside. Maybe that's the female part. I don't know. But if you have only gene B active, the leaf-like structure turns into a sepal. If you've got A and B together, it becomes a petal. If you've got A and C together, it becomes a stamen. If you've got only C, it becomes a carpel. So that's just talking about how different genes and different combinations will produce different things. And we'll apply that same idea to Hox genes now. Let me stop for a second. And I feel like that clock and wave model was kind of complicated. Let's see your faces. Give me some thumbs up and thumbs down. How many people follow that? Or do you want me to go through it again? Angelique, Stefan's up and up thumb. Michelle, Simmons came and went. Elijah's up. You've got a little reaction thing here too. Can you give me a thumbs up, thumbs down on that? Try to get feedback here from people like Sammy Robinson, Alex McKieran, Nishi Kenderwal. Okay, so Asha Gupta's in. Rocky's ignoring me. I'm kind of used to that by now. Okay. All right, so let's move on and talk about Hox genes then. Hox genes. Okay, so now we're getting into why, even though this is pre-Semitic mesoderm, and it looks like any pre-Semitic mesoderm, if you cut it out and put it in an earlier place, in a younger embryo, you can see that it's already patterned, right? So this is already patterned to become thoracic, even though there's no thorax right now, right? But those genes have already been active. That makes it like that. So what genes are there that make it do that? It has to do with these Hox genes. So you've got 4 different, what do we call them? I'm sorry, I just want to get the name right before I mess it up and have to come back on Wednesday and correct it all. These are called paralogs. So this is a set of, looks like 11 genes. There's like 13 possible genes, but of each paralogous group, some of them are missing some of the Hox genes, okay? So you can see for Hox gene 9, all of the paralogous gene clusters have got number 9, but for some of them, like the Hox gene B, doesn't have 10, 11, or 12. So anyway, each of these is called a gene cluster, 1 through 13. And come on, you. Oh, what am I doing? Trying to get this thing to there. Okay, so each of these is a gene cluster. Cluster A, B, C, and D. A is paralogous to B, is paralogous to C, is paralogous to D. So paralogous means coming down this way. So each of these genes will get expressed in the sequence that they are on the genome. This is really weird. This is the only set of genes that get expressed like this. But this is the 3' end of this gene cluster, and this is the 5' end of the gene cluster. Gene number 1 will be expressed first. Gene number 2 will be expressed next, then 3, then 4, all the way down to 13. So they're expressed in the order that they're sitting on the genome, which is pretty weird if you ask me. And all the 1s will be activated at once, all the 2s activated at once, all the 3s, 4s, and 5s. As you go down, they're all the paralogous genes are activated at the same time, sequentially, you might say. So 1 and 2 are in the hindbrain, like the first somites that get formed, and the last ones are down in the tail, which makes sense because it's happening during neurulation as they're coming down. Now, getting back to the combinatorial thing, if you got all the 1s being expressed, that ends up being hindbrain up here, okay? All the 2s being expressed, it ends up a little bit further back. The A's produce this area. The 4s are down here. So they're getting expressed from head to tail in a sequential manner. And you've got, in terms of combinations, the 1s get turned on first, but they never go off, okay? So if you have only the 1s on, it ends up in the hindbrain. But that's going to stay on, and eventually you get the 2s and 3s to turn on, and that ends up in the cervical area. So you've got all of these genes on at once. And as you go further and further back, you get all the way to the tail and all the genes are on, and 13 just turns on for the tail section, okay? So position in the body is based on how many of those Hox genes are turned on. At the beginning, you've got only the 1s, and towards the end, you've got all of them on, and 13 coming on last. And overlapping, oops, shoot. So each region of the mesoderm is determined by a specific set of Hox genes in combination. So here's your thoracic. Thoracic's got all of these genes on at once. The ones that are just coming on, by the way, keep this in mind too. The ones that just come on in that segment are at the highest level. So if you look at B1, it's highest when it's the only one on, but towards the back, you've got less and less B1 being expressed. It's still there, still being used in combination, but there's less of it than there was right at the beginning. So each one peaks when it first gets turned on, and then it becomes less and less. Okay, interesting thing about this too, is when you look across species, the same genes are turned on for the same regions, even if the regions are different sizes. So a chicken neck is really long compared to a mouse neck, right? So you figure your mouse vertebrae are going to be in here. Your mouse cervical vertebrae are up in here, okay? And these are due to genes of 4 through 5, okay? And here you've got a lot more somites for the chick cervical vertebrae, but it's the same genes in the same positions being turned on. Now, when you get into the thorax, the chicken's got a shorter thorax than the mouse. So we've got more somites in the mouse and fewer in the chick, but it's the same Hox genes being turned on in the same relative positions as you go down. And the same thing with this, and then the tail section as well, okay? So it's the combinations of those Hox genes that determines what that structure is going to be. And that's why in this pre-Semitic mesoderm, those Hox genes have already been turned on. So if you take it out and flip it around, it ends up upside down because you've already got the sequential activation of those Hox genes turned on. Excuse me. I don't know why. Whenever I zoom, I burp all the time. Okay, hang on a second. So this is how it works. So this next slide is a PowerPoint animation that runs automatically. What I want you to watch as we do this is the node keeps going backwards slowly, okay? And like we saw in the last thing, as the node is moving, somites are being made, right? because of that clock and wavefront idea. Now, the other thing you're going to see is Hox genes starting from the posterior end of the embryo in a wave from the back to the front. So you'll see Hox gene 1 turn on in the posterior cells, and that wave flow all the way up past the node and then get incorporated into the somite. And then the wave keeps moving, your next Hox gene gets turned on, and as the somite gets formed, it gets incorporated into a somite. And so as that node keeps moving, whichever Hox genes are on when that somite gets formed are going to determine the position of that somite in the body, anterior, posteriorly. Let's watch that. Okay, node going back, Hox1 being expressed. First somite gets laid down, Hox gene 1 is expressed in that somite. Here comes number 2, so really 1 and 2, that gets put into a somite. 3 coming through, and then 4, and then all the way through 13. So now these somites then are patterned based on which Hox gene was flowing through when it differentiated. Okay, and again, remember, this has got only Hox1. When you get down here, you've got all the Hox genes on, including the newly expressed ones. Okay, so why does this happen in waves like this? Why do they happen sequentially like this? Well, as in everything else, it's laid down in your DNA, right? It's all the computer program. So what happens is cells get hit with some Wnt, get hit with a Wnt signal, right? Wnt's pretty common here in our cells. When it does, it turns on that first Hox gene. Okay, that first Hox gene then causes transcription of CDX2, another transcription factor. CDX2 then starts to activate the next Hox genes in sequence. And that kind of feed forward effect goes all the way through to your last Hox gene number 13. Hox 13 then turns off CDX2, so that shuts off all the other genes. So at that point, the whole thing stops. So the first genes get turned on, the first genes turn on the next genes, which turn on the next genes, which turn on the next genes, all the way down the line till the 13 gets turned on, and that turns all the rest of them off, and the whole process stops. That, my friends, is pretty complicated stuff, if you ask me. I have not gotten any questions from anyone, though. Everyone's fine with it. Geez, Dr. Slish. Nishi's following. Good. At least somebody is. Okay, so that whole spiel took me about 20 minutes to tell you. Took me about maybe 10 hours to figure out on my own. So I know it's complicated. And maybe you're a little overwhelmed right now, which is why you're not asking questions. Maybe I just explained it really, really well because I am pretty awesome. Can I get a little feedback from you guys? Are we following? Are we not following? Hopefully you guys are studying because we do have a test next Monday. Monday. Can I get some thumbs up, some thumbs down? I think there's a middle finger there in the reactions too, which you're welcome to give me if you want. I'm okay with that. So HOXA blocks Hox genes. Let me answer that in a second. Getting thumbs up, thumbs up. Okay, so HOXA blocks Hox genes. No, the way you want to look at it is HOXA1 is going to be turned on first, as is HOXB1, HOXC1, HOXD1. They get turned on first, and they're going to activate later ones. HOX13, the very last one, whether it's A, B, C, or D, that's going to shut the other Hox genes off. Is there a video on Hox genes? I'm sure there is. Tell you what, let's put that out there for 3 bonus points. Whoever comes up with the best Hox gene video, as determined by me, will get 3 bonus points, and I'll post it up in Moodle so everyone can see it. Love that. Stefan's probably already got one. Okay, any questions or comments based on all of that discussion there? Stuff? Nothing? Nothing. See, this is what I hate about Zoom. It's just so unresponsive. I'm just sitting here in a room. Look at that. I look better this way. Just sitting here in a room talking to myself. Tyler Montano's on it. You crazy. Easy 3 points. Okay, hang on. I will decide if you get the 3 points. Remember who's in charge here. Let's watch this.
Animal body plans. Animals have a variety of body plans. Flies have 6 legs, mice have 4, and millipedes have many. Scientists have proposed that changes in a group of similar genes called the Hox genes have created the diverse animal body plans we have today. To test this hypothesis, our lab is focused on crustaceans. Crustaceans are an excellent group of animals for studying body plan evolution because they have many different body plans.
Nice thing about crustaceans too is after you've done your experiment, you can eat your experimental animal.
By examining Hox gene
Cocktail
crustaceans,
sauce.
we can understand how these genes have changed to produce new body plans. In particular, our lab focuses on the sandhopper. Let's stretch out the sandhopper body plan so we can see each segment. The sandhopper has antennae, many mouth parts, claws, forward walking legs, reverse walking legs, swimming legs, and anchoring legs. First, we figured out where each of the Hox genes is turned on. Then we deleted each of these Hox genes to see how this would affect the animal's body plan. For example, if you delete the Hox gene called abdominal A, the animal's backwards walking legs turn into forward walking legs, and the animal's swimming legs turn into anchoring legs. This means that abdominal A is necessary for making backwards walking legs and swimming legs.
And you can kind of see based on what I said, you would expect these genes to be on back here as well, right? You would expect the genes that made this to be on here plus an additional gene. You take out that additional gene, all you have is these genes, you get the same structures here.
The portion of the body that turns on the abdominal A gene has changed over evolutionary time. If we look at other types of crustaceans, such as shrimp and pill bugs, we can see that the part of the body that turns on abdominal A has changed. This change leads to a difference in the body plan of these other crustaceans. Currently, we are starting to determine what is responsible for the changes in where abdominal A is turned on.
Okay, there's your Evo-Devo, evolution and development. Small changes in evolution can have a big developmental difference and cause evolution of the species. Okay, I have to admit, this isn't exactly the Hox gene I was hoping for. Let's see what our friend Kwame has to say.
My name is Zifan Patel. I'm a faculty member.
Okay, this is probably good, but it's 35 minutes, so I'm not going to watch it now. Let's see who can come up with the best video that is, email me the best video that is, here are the parameters. The parameters has to be less than 5 minutes, and it has to be chick. Go. And I will talk to you again on Wednesday, and we will see. Okay.
Hey, Donald.
Later, babe. Did I say later, babe? Did I just say later, babe? My God. It's like I'm sitting in a room all by myself. All right, later, guys.